Hi everyone, welcome back for lesson 4.9 on mutations. As always, make sure to fill out your guided notes and keep up with your vocabulary as we go through this lesson. So here we have a few vocabulary terms. First off, we have a mutation, which is the change in the genetic material of a cell. We're going to talk about two basic types, a point mutation or a chromosomal mutation. Point mutations are gene mutations in which a single base pair in DNA has been changed. Chromosomal mutations are mutations that affect whole chromosomes and whole genes rather than just individual nucleotides. So first we're going to look at what we call gene or point mutations. So again, gene and point kind of interchangeable, but for vocabulary's sake, we're going to stick with point mutations. So the first one, we're going to be looking here at these examples, but here we have the before mutation sequence, your original DNA changed to RNA, which is then here into your amino acid. So kind of keep this as a reference. So for a substitution, that is one base is changed to a different base. So here's our first one. Here'd be a substitution. So that original sequence was actually GCA. Now we see that that T was substituted for the C, and that's going to end up changing the amino acid. So we had ARG, ARG in our original one. Now we change that to HIS. So that's an example of a substitution. They usually impact only one amino acid, but there is something called a silent mutation in which the substitution did not change the amino acid. So kind of pause and think to yourself, how could a silent mutation occur? And a hint is, look at your codon chart. So if you're looking at your codon chart, you remember that there are many possible codons that could code for ARG, right, ARG. So for this one, say that this was changed, however, this specific codon still coded for the same amino acid, that would be the silent mutation. Other two types, we have what we call frame shift mutations, and those are mutations that shift the entire reading frame of a genetic message by inserting or deleting a nucleotide. So our two types of frame shifts are insertions and deletions. Here's an example of an insertion. An A was added, and then you will see that actually that whole amino acid sequence was now changed after met or start, from the original. And then deletions as well. So here this G was deleted, same thing, the whole what we call reading frame will shift, therefore our whole amino acid sequence will shift. So you will see that generally the frame shift mutations are going to have more of an effect on the entire amino acid sequence than just one substitution. So then we also have our chromosomal mutations. So there are 4 different types. And remember, these are affecting whole chromosomes. So even though we have here things like deletion, remember that's different than this deletion, right? Because this is referring to one base. This is referring to a whole piece or part of a chromosome, So that's the difference between that gene or point and chromosomal mutations. So our 4 types are deletions, duplications, inversions, and translocations. So deletion, loss of all or part of a chromosome. Duplication, we're going to have an extra copy of all or part of a chromosome. Inversion reverses the direction or parts of a chromosome. And translocation, part of a chromosome breaks off and attaches to another. So that's that image here. So here are a couple examples. This is some extra information. You don't need to memorize this, but just some examples that are caused by single genes. Sickle cell and Huntington's, both are caused by single gene mutations. So if you'd like to read more on that, please feel free. So then we have our effects of mutations. So genetic material can be altered by natural events or by artificial means. Many mutations are produced by errors in genetic processes, and mutagens are chemical or physical agents in the environment that can interact with DNA and may cause a mutation. So again, this would probably be that artificial means. We have some examples such as chemical and physical mutagens. The effects of mutations on genes vary widely. Some can have little or no effect, and some produce beneficial. Some negatively disrupt gene function as well. So then we can also have some changes in our chromosome number, and that results sometimes as an error in meiosis. And remember, we're talking about meiosis here because we're referring to the production of gametes, right? So production of gametes, meiosis. So non-disjunction is the error in meiosis in which the homologous chromosomes fail to separate properly. So here we have your original cell, non-disjunction occurs, okay, and that can lead to an abnormal number of chromosomes, and the most common of which are what we call trisomies or monosomies. So a trisomy is a condition in which an individual has 3 copies of a chromosome, and a monosomy is a condition in which an individual has 1 copy of a chromosome. So here are some examples. Okay, we looked at karyotypes in that last lesson. However, okay, here if we look, this one has 3 chromosomes at number 21. Okay, so there are some examples, but there are many human syndromes related to changes in chromosome number. We have our autosomal. Okay, we have examples of Down syndrome and Edwards syndrome. So trisomy at 21 here, this would be what a person with Down syndrome, their karyotype would look like. We also do have some errors with our sex chromosomes, okay, possibility of the changes in chromosome number for those two. Common ones include Turner syndrome, which is a monosomy of the X chromosome, and Klinefelter syndrome, which includes an extra X chromosome. And then we also have polyploidy. When a complete set of chromosomes fails to separate during meiosis, the resulting gametes are triploid or tetraploid. And triploid, again, would just mean 3. Okay, tetraploid would mean 4. So here is your polyploidy definition, condition in which an organism has extra sets of chromosomes. So polyploid plants are often larger and stronger, and sometimes polyploid plants are seedless. Okay, so here is a check for understanding right before your exit ticket. So go ahead, pause here, try this on your own. Okay, so number 1, again, we are looking at chromosome. Okay, so several genes from chromosome 3 are detached and reattached. That would be a translocation. Number 2, third copy of the 21st chromosome, that would be a trisomy of 21, which is also known as Down syndrome. Number 3, okay, if we have a codon change, right, we have A now turned to U, so that would be a substitution, and again, that is a type of point mutation. Number 4, okay, we have our codon originally AAA, okay, it is now inserted, right, a U is inserted, so that would be an insertion, and specifically if you wanted to put like frame shift insertion, that's also fine. And then number 5, okay, we have an AAA, and that was changed to AAG. For this one, okay, we know it's a substitution because the A turned to G, but if you looked at your codon chart, it's actually a silent mutation, and that's because lysine was AAA, and AAG is also lysine. And then number 6, so the silent mutation would most likely cause the least dramatic change because the amino acid sequence did not change. And we have reached your exit ticket, so go ahead and pause here and try this on your own. Okay, so number 1, a single nucleotide base pair would be a point mutation. Number 2, okay, a single base pair in which a new base pair is added, again, because we're looking at base pairs, okay, so point mutation, that would be an insertion or frame shift. Number 3, chromosomal mutation, in which part of the chromosome is lost, that would be a deletion. And again, you probably want to make that distinction that we are referring to a chromosomal deletion, not a deletion like a point or frame shift. And then last one, number 4, a single base pair that does not change the amino acid, that would be a silent mutation. If you also put substitution by that, that is also fine. So that is the end of lesson 4.9. Thank you for watching. Please check your notes and vocabulary and reach out with any questions.